I only find things on gluconeogenesis, but I do already understand beta oxidation.
Wait, what are you talking about?
Girl, weren't you in class when she was giving us these scenarios for lecture?
I was in class, but I didn't take any scenarios. I was talking too fast. Me studying me.
Well, the first one was about gluconeogenesis and how beta oxidation works during exercise.
And well, the other one is why our body chooses to use glucose instead of lipids during exercise.
Oh, and don't forget the maintenance thing. Something with explain diseases and linked to exercise metabolism.
But I don't understand beta oxidation, so now I understand.
Okay.
Hey, I have to play teacher game or but on small.
Teacher game.
So Beta, you're not serious?
Yes, calm down. Okay, this is how I thought about it, right? Since he understands gluconeogenesis and she understands beta oxidation, and I can understand some things, right? You know, we can go up and be like, okay, I'm a teacher and teach you class and teach us like beta oxidation and gluconeogenesis. So we just put your teacher roles to explain each other and we'll understand that way.
Alright, well, let's try that.
Well, I'll be first. I'll be Mr. Gluconeogenesis.
As usual, Miss Fish.
I'll be explaining gluconeogenesis. Gluconeogenesis occurs in the fasting state, right a little before glycogen stores are used up in the liver. This is an energy-dependent process to make glucose internally. Gluconeogenesis is not the exact reverse of glycolysis because there exist three unique, irreversible reactions of glycolysis, and so gluconeogenesis has to find a way to strategically bypass these reactions. It does that by changing three enzymes for three more energetically favorable reactions. The first bypass reaction is where pyruvate is converted to oxaloacetate in the mitochondrial matrix by pyruvate carboxylase. Once oxaloacetate is formed, it cannot leave the mitochondrial matrix and is therefore reduced to malate by mitochondrial malate dehydrogenase. Malate crosses the mitochondrial membranes into the cytosol where it is oxidized to oxaloacetate by the enzyme cytosolic malate dehydrogenase. Oxaloacetate is therefore converted to PEP by the enzyme PEPCK, and PEP re-enters the gluconeogenic pathway. The second bypass reaction is the conversion of fructose 1,6-bisphosphate to fructose 1,6-phosphate by the enzyme fructose 1,6-bisphosphatase. This is the regulatory enzyme for this reaction. The second bypass reaction is also a hydrolysis reaction where an inorganic phosphate is formed. The third bypass reaction is the hydrolysis of glucose 6-phosphate to glucose and an inorganic phosphate by the enzyme glucose 6-phosphatase. This overall reaction utilizes 4 ATP and 2 GTP. And this is because there's one other reaction that utilizes ATP, which is the conversion of 3-phosphoglycerate to create 1,3-bisphosphoglycerate by the enzyme phosphoglycerate kinase. But the processes of gluconeogenesis and glycolysis are hormone regulated by insulin and glucagon. Glucagon, the polypeptide hormone, increases the activity of fructose phosphatase 2, which breaks down the allosteric inhibitor fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is the inhibitor of the regulatory enzyme fructose 1,6-bisphosphatase. Glucagon allows for the stimulation of the metabolic pathway gluconeogenesis, increasing the substrate availability such as glycerol and activating its regulatory enzyme.
I'll be explaining the beta oxidation pathway first. Does anyone know what I'm talking about?
No.
I'm very lost.
I'm confused. I'm very confused.
Well, beta oxidation involves the catabolism of fatty acids in the mitochondria, where every cycle 2 carbon fragments are removed. Hello. You see me, Jim?
I just keep in track of what you're talking
Anyway, this pathway produces acetyl-CoA, FADH2 and NADH. Now after a long chain fatty acid enters the cell, it is converted in the systole to its CoA derivative by the enzyme. Anyone else?
No. Does it have to do something with long chain?
Yes.
Okay, well, I don't know the rest.
Well, the enzyme is the long chain fatty acid CoA synthetase, also known as thiokinase. Now the next step is the transport of the CoA derivative into the mitochondrial matrix, but the inner membrane is impermeable to CoA. So the carrier carotene molecule is used and transports the long chain fatty acid CoA into the mitochondrial matrix. Now acylcarnitine is made by the action of the enzyme CAT1 and is located on the outer side of the inner membrane. And acylcarnitine is now free to move from the systole into the mitochondrial matrix by the antiporter protein, where at the same time the carnitine group is removed by the enzyme CAT2 and shuttled back out, and the remains of the fatty acid carnitine reacts with CoA to produce fatty acid CoA. Okay, class, I have a few questions. What is the other name for CAT2? 1. My bad. CAT1.
I can make a mistake and answer my question. So,
Yes.
Jimon, wait, that wasn't this paper.
I think so.
Oh, CAT1. Is it CAT?
Is it CPT1?
Yes, you're correct. And also, could you state 2 sources of carotene?
I don't know that.
Is it from your diet, like meat?
Yes, that's one. And the second one?
Is it amino acids?
Yes. How much?
I think it's 2, isn't it? From like
One is lysine.
Yes.
and methionine or something like
Yes, that's correct. Also, could you tell me when beta oxidation will occur? Because just because there's fatty acid available doesn't mean it's going to happen.
Well, I think beta oxidation occurs when the energy charge in the cell is low.
Yes, that's correct. Okay, now we go on to the interesting part. Now there are 4 reactions that the fatty acid CoA undergoes. So the first reaction is the oxidation reaction catalyzed by acyl-CoA dehydrogenase. The second reaction is catalyzed by an enol-CoA hydratase, and it's a hydration reaction. The third is another oxidation reaction catalyzed by 3-hydroxyacyl-CoA dehydrogenase. And the third reaction is a thiolysis catalyzed by the enzyme beta-keto-acyl-CoA thiolase. Now, with that being said, now my question is, how does beta oxidation link to exercise?
Miss, before you continue, in these reactions, right, is it that NADH2 and FADH2 and acetyl-CoA is being produced in these reactions?
Yes, you are correct.
Okay, and where in this reaction is it taking place? Like in the first 2 oxidation, like the first oxidation, NADH is taking place.
Right.
Then in the second oxidation, which is the third reaction, FADH2 is being produced.
Right.
And then at the end, we have acetyl-CoA
Right.
being produced.
Right.
Right? So, and you said that it's a highly energetic pathway, right? So is it that these things like interact with the TCA cycle and goes to the TCA cycle to an oxidative phosphorylation to produce ATP? So how does this link to exercise? Well, I think me and Domi know the answer because we did our research and you want to go on and help them?
Sure, no problem. Sure. Hold on.
Yeah, very well. Me and Dominic is going to explain how the interaction of beta oxidation and gluconeogenesis during exercise. Right? So, who wants to go first?
Well, I'll go first since my one is in a video format. So I'll just press play and you guys will see it, okay?
How can gluconeogenesis and beta oxidation interact? Well, it is proven by the Randall cycle, which simply states that increased fatty acid oxidation causes a decrease in glucose oxidation. This can lead to glucose uptake declining and hyperglycemia. This cycle has been used to explain lipid oxidation and the sparing of glycogen utilization in muscles during exercise. In the cycle, beta oxidation increases by the entry of fatty acids into the cell. Since beta oxidation is responsible in breaking down fatty acids to acetyl-CoA, acetyl-CoA will increase, and this will lead to the inhibition of PDH. And therefore, pyruvate is rerouted to gluconeogenesis. Since the pyruvate cannot be broken down to acetyl-CoA and will be converted to glucose. So then, what can overrule Randall cycle? Well, AMPK can. The activation of AMPK depends on the duration and intensity of the exercise, which will cause oxygen deprivation. The AMPK regulates energy balance by turning on catabolic ATP generating pathways, which are fatty acid oxidation and glycolysis, while switching off anabolic ATP consuming processes, which are lipid and protein synthesis. In summary, exercise can affect the Randall cycle and AMPK activation.
So I'm going to be explaining how gluconeogenesis will get exercise, since Dominic just explained the interaction between beta oxidation and gluconeogenesis. Well, as Javon mentioned, gluconeogenesis occurs during the fasting state, right? But during the exercise, it occurs during prolonged exercise, like after a little while before glycogen source has been used up, right? So, apart from glucose, there are three major precursors that gluconeogenesis can use to retain back to glucose, such as lactate, glycerol, and amino acids, right? However, these three main precursors are influenced by the intensity of exercise. So, for example, if the intensity of the exercise is just below lactate threshold, which is before lactate is increasing in blood, right? Glycerol will be influenced and it will go through that pathway as seen on the board, right? However, if lactate threshold is exceeded, that is the lactate concentration in blood increases, then lactate from the muscle will be sent to the liver, right? And then in the liver, it goes to the gluconeogenesis pathway, which produces glucose. This glucose then is sent back to the muscle and is utilizing energy. This cycle that I just explained as a cycle is known as the Cori cycle. So, since we and Dominic just explained beta oxidation, gluconeogenesis, right? Which one you think that during exercise we will be utilizing more? Fats or carbohydrates? Fats? No, that's kind of wrong, right? But I would say it's carbohydrates. Why? Because carbohydrates can be used with or without oxygen, and during exercise you will be going through anaerobic respiration in and out, right? Also, carbohydrates releases energy much more faster, right? And you need carbohydrates to be present in order for fat to be utilized, right? And well, they are the sole source for energy for the central nervous system. So this is why carbohydrates is used instead of fats during exercise. So that's basically how gluconeogenesis, beta oxidation, and stuff works in exercise. So yeah, that's just about it, short and sweet.
Not bad at all. You guys have been practicing talking. You can have a seat. I'm glad to see all of you so early. So what have you all been doing so far? What have you gone through?
Well, we have gone through how beta oxidation and gluconeogenesis interact during exercise. So yeah, that's just basically it. We just went through the gluconeogenesis pathway, beta oxidation pathway, the importance, and how to recognize it.
Okay, cool. Is there anything you all didn't understand?
Well, we didn't get to reach the disease part, and nobody had no notes on it, so I basically guess that's what we didn't understand, the disease part.
Alright, that's no problem. We didn't even reach that far, so we're going to hit that today. Right? There's a couple of diseases, but we're going to focus on the enzyme CPT CPT2 deficiency. So what's CPT2?
CPT2 removes carnitine from fatty acid carnitine to reform fatty acid CoA.
Alright, sorry. If that doesn't happen, right? What's going to happen to the beta oxidation? What's going to happen then? If CPT2 isn't functioning properly?
If CPT2 isn't functioning properly, then the carnitine from the acid carnitine will be able to shuttle back across into the system. So therefore, beta oxidation will be hindered in that it cannot form that fatty acid.
Okay, I think I know what you're trying to say. Alright, that's good. Good try. So if all this happening, right, and beta oxidation isn't occurring properly, what's going on with your body? What's going on with your muscles?
Like you might get muscle fatigue and stuff like that.
Alright, you'll get muscle fatigue, you'll get weakness, sometimes you might even get muscle pain, almost debilitating, right? So, if that's that, right, now we're going to look at the treatment, right, of this. Now, if CPT2 isn't functioning properly, right, you could wonder why sometimes it's a deficiency, right? What you can use, right, is medium chain fatty acids, right? This is because medium chain fatty acids don't need the CPT2 enzyme, right? Or you can use L-carnitine. What doctors usually do, they use L-carnitine, and they will go through a diet plan with you so that they can see, okay, no long chains here, no long chains here, medium chains here, so on, right? So that's basically it. That's all you all need to know for the diseases part, because that's basically what we narrowed down to. Now, as you can see, next week we're doing ketosis and acidosis. We're not really going to focus on how it's done. That's another bad lecture. What we're going to do is just we're going to do a disease again, right? Another disease, right? Which is acidosis. We're going to see how that happens. And all that already happens here is acidosis is like a aftermath of ketosis. When these ketone bodies form and your blood takes in these ketone bodies and it can't come out or there's too much, your blood pH it lowers and you just happen to get acidosis. It's just kind of detrimental because your blood sees these ketone bodies as a poison. But anyway, that's it for class today. Sorry, I'll see you next week.
Okay.